very special process of cell division called meiosis. Now, before we jump into this, you need to understand what the whole thrust of this goal of this division is, and that is to make the sex cells. So, first question. If the job of meiosis is to make the sex cells, the sperm and the egg, then the question is, where is this happening? What organs are actively doing this process? And the answer is the testicles and the ovary. Those are the only organs that will do meiosis. And even then, most of the cells within those organs are not doing this process. It is only the ones that are actively engaged in making these sex cells. Gamete is a blanket term that refers to both sperm and egg. So the goal of meiosis is to make the gametes, to make the sex cells. Second question, how are the sex cells special? The answer is the sex cells were special because they have half the normal amount of DNA. They have to have half the normal amount because the egg has 23 chromosomes and the sperm has 23 chromosomes. And when the sperm and the egg unite, those 23 separate chromosomes turn into 23 pairs of chromosomes. So we've got to take a normal cell that has the normal amount of DNA and turn it into one of these sex cells that has half the normal amount. One problem? The problem is because this is based off of mitosis, the cell that begins meiosis starts off the same way. And remember, the cell that began mitosis didn't have one complete set of chromosomes. It actually had two complete sets. It didn't have $1 in it. It had $2 in it. The cell that begins meiosis also has $2 in it. We want to end up with a cell that has half the normal amount. Mitosis, I want to end up with cells with the normal amount. So my cell with $2 in it splits, and I end up with two cells that have $1 each. Meiosis, I don't want to end up with cells that have $1 each. I want to end up with cells that have half the normal amount. So I want to end up with cells that have how much in them? 50 cents each, half the normal amount of DNA. If I start off with a cell with $2 in it, after the first division, I've got two cells that have $1 each. Now what do I have to do? I have to divide those again. I take those two cells that have $1 each, I divide them another time, and then I end up with four cells that have 50 cents each. It follows the exact same basic steps as mitosis. It is divided into two separate steps, meiosis 1 and meiosis 2. The very short definition of meiosis is it is just mitosis twice. I do prophase, metaphase, anaphase, telophase once, and I start with one cell and end up with two. And then I do prophase, metaphase, anaphase, telophase a second time, and my two cells have now divided again into four cells. All right? That is the short definition. It is a little different than that. It is a little more complicated than just mitosis twice. But that is the Cliff Notes version of what meiosis is. What happens in the steps are the same things that happen in the steps of mitosis. In prophase, the chromosomes wind up. In metaphase, the chromosomes line up. In anaphase, the chromosomes get separated. And in telophase, the chromosomes will unwind, and then eventually the cell will split in two. And then you just do that process twice. The difference happens in Anaphase 1. The difference in Anaphase 1 is, if you'll remember, in Anaphase, in mitosis, I physically ripped that chromosome in half and drug half of the chromosome to the top and half of the chromosome to the bottom. That's not actually how we do it in Anaphase 1 of meiosis. In Anaphase 1 of meiosis, what I actually do is I separate the pair. I don't separate the chromosomes. All right, let's follow it through. All right, so I begin prophase 1 of meiosis 1. The red chromosomes are moms. The blue chromosomes are dads. The chromosomes wind up during prophase, and they line up along the equator. Notice that they kind of line up a little bit different. instead of wires going from the top and the bottom to each chromosome, a wire will come from the top and attach to one of the pair, and another wire will come from the bottom attached to the other of the pair. And so when I go into anaphase, I have not ripped the chromosomes in half. I've just pulled one copy of the pair, one half of the pair down, and the other half of the pair up. Once I go through prophase, metaphase, anaphase, telophase 1, now it's time to start prophase, metaphase, anaphase, telophase 2. Here's my one cell that's about to split in two. I've now split it in two. The chromosomes will line up in the equator again, and now look how the spindle apparatus, the wires are attaching one wire from the top, one wire from the bottom. Every single chromosome now has a wire coming from both sides. And so when these two cells pull, they're going to actually rip those chromosomes in half, and when they rip those chromosomes in half, I end up with 1, 2, 3, 4 cells. We have not quite finished cytokinesis in this particular picture. There are still technically 2 cells. They will become 4 cells once they finish. But remember that the whole process is I started with a cell that had 2 copies of every chromosome, or rather, 2 complete sets, 4 copies of each chromosome. And what I want to end up with is I want to end up with a cell that has 1 copy of each chromosome in it, which is half the normal number. So the only way to go from 1 cell with 4 to 4 cells with 1 is to split a first time and then to split a second time. Meiosis 1, meiosis 2. This shows a comparison between mitosis and meiosis. Mitosis is the one we understand best. Here's the cell that's going to replicate its DNA. It's going to take and make a copy of each of its chromosomes. This species only has 2 chromosomes. Chromosome pair number 1, chromosome pair number 2. So what it's going to do is it's going to make a copy of it, and so I now have 2 of my big red ones and 2 of my big blue ones, 2 of my little red and 2 of my little blue. It's going to go through this process called mitosis. It's going to split, and the chromosomes are going to rip in half, and I'm going to end up with 2 cells that look just like the parent. All right? I do a similar step in meiosis. When I split them the first time, I don't actually rip the chromosomes in half like I did here. I just split the pairs. So all of the red one goes one way, and all of the blue one goes the other way. And then I split those again, and I end up with 4 cells that only have 1 of the big chromosomes. The normal number is to have 2. And then only 1 of the little chromosomes. And again, the normal number is to have 2. All right? So I start off with 1 cell that has 4 of each chromosome, and I end up with 4 cells that have 1 each. And if this is a man, all you have to do is put a tail on that, and I've now made sperm cells. Women do it a little differently. They're a little more complicated, but this is how meiosis is accomplished in men. Remember, the only organs that do meiosis are the testicles and the ovaries. Those are the only ones. The product of meiosis in the ovary is an egg. And the product of meiosis in the testicles is a sperm cell. The whole point of these 2 cells is to do what? To come together to make a new life. And if the new life is supposed to have 46 chromosomes, that means that each sperm and egg has to have half the normal number. Not 46, but 23 and 23. So when I add the 2 of them together, I end up with 46. I end up with the normal number, the normal amount. In that first picture that we looked at of meiosis, you may have noticed that some of the chromosomes appeared to have extra bits. Some of the red chromosomes had little blue bits on them, and some of the blue chromosomes had little red bits on them. This is a phenomenon that happens during meiosis, only during meiosis, and that is that chromosomes will sometimes exchange legs. And this is called crossover. So this is mom's chromosome, this is dad's chromosome. These 2 chromosomes are getting ready to go through meiosis, and you'll notice where they'll cross over and they'll exchange legs. And so that this chromosome right here is now not 100% just moms. It's part moms and part dads. And the same thing for this one is true. It's part dads and part moms. The whole reason behind this goes back to the whole reason behind sexual reproduction in the first place, and that is to increase variety. Variety is what makes life so successful. The reason why we do sexual reproduction is to increase variability. Because the world is ever changing, and since the world is always changing, the organisms that live on it must change with it. Because the organisms that live on the planet must change with it, you've got to have new combinations of DNA. The only individuals that are going to leave behind babies tomorrow are the ones that can survive today. But if tomorrow is different from today, then that means that the ones that survived yesterday might not be fit to survive the new conditions of today. That's what variability is all about. So variability, there are actually 3 different methods by which, during sexual reproduction, we increase variability. And like I said, the first one is crossover. The second one is called independent assortment. Independent assortment says there's no reason why the chromosomes line up in the order that they do during metaphase. If I were to go back and label these chromosomes, don't have a good picture of it. If we were to go back and label these chromosomes and we see how they have lined up, each time you run this process called meiosis, they will line up in a different order. Imagine I have a magic microscope that allows me to pause cell division. And when these chromosomes line up in a certain order for meiosis, and I number them, I give them a number, 1 through 46, and then I'm able to rewind this cell and take it back before prophase, and then press play again, and let it to come forward up to metaphase again. Each time it comes up into metaphase, those chromosomes will line up in a different way, in a different manner. Independent assortment. Each time the chromosomes line up, they line up in a different way. And what that does is it makes sure that I don't always make sperm cells that are the same combination of chromosomes, and so that the egg cells not always the same combination. Remember, what's the whole point of sex to begin with? And that is to mix up the DNA. So independent assortment mixes up the DNA. The last one is called random fertilization. In random fertilization, this shows independent assortment. They line up in a different manner each time, so you end up with different combinations. The last one is called independent assortment. Random fertilization. And random fertilization means 2 things. The first one is, there's no reason why the egg that was released this month won. There are about 20-ish thousand eggs inside of a woman's ovaries. If there's about 20,000 eggs in a woman's ovaries, then one of them gets chosen to get released each month. Just one. Why did that one get chosen? And the answer is there's no good reason why. It just happened to win. Well, different eggs will end up with different combinations of genes in them, different combinations of chromosomes in them, and so each one is going to be different. Random fertilization. The first meaning is there's no reason why that one egg was chosen to be released this month. Second is, a woman releases 1 egg a month. How many sperm cells does a man release in 1 ejaculation? And the answer is, well, it depends, but average is about 300 million. Winning a 1 out of 20,000 lottery is a rare event. How many sperm cells make it into the egg? And the answer is 1. Winning a 1 in 300 million lottery is huge. Why did that 1 sperm cell get in? And it's not because it was the fastest. That's not the reason why. It actually takes the actions of thousands of sperm cells to be able to get 1 inside. The egg has a very tough coating around the outside of it that literally has to be dissolved. And it takes the actions of thousands of sperm cells actively attempting to dissolve that coating to be able to get it thin enough so that 1 cell can work its way through and to get in. Why did that 1 get in? there's no good reason why that one got in. That's the second definition of random fertilization. So, random fertilization, two definitions. No reason why that egg was released. It just won. It just got lucky. No reason why that sperm cell got in. It just won. It just got lucky.